My name is Sylwia Bobis-Wozowicz and I work at the Jagiellonian University in Kraków, Poland. The main focus of my research is regenerative medicine. I try to combine cell-based and gene therapies for the purpose of developing innovative, safe and efficient methods for gene modification which can be used for the treatment of genetic diseases. In my work, I utilize small, naturally produced nanovesicles, which are called extracellular vesicles, or EVs in short, for the delivery of genome editing tools directly into a living organism. And these tools, including the most commonly used nowadays systems, the CRISPR-Cas9, act as molecular scissors and cut the DNA at strictly defined genomic position. This allows for precise genome modification, which is necessary, for example, to correct a genetic disease. The major advantage of our system is its safety and efficiency. We were able to achieve high genome editing rates, particularly in the liver of the experimental animals. Therefore, our system may replace the existing technologies based on viral vectors, which use is often associated with harmful side effects. On the contrary, we use natural nanovesicles, which are produced by cells and therefore are well tolerated upon in vivo injection. What is also important that the vesicles themselves provide excellent protection for the genome editing tools from their degradation after in vivo delivery. Importantly, we use stem cells-derived EVs, including mesenchymal stem cells or induced pluripotent stem cells, which are both used in medical applications. And therefore, our technology provides a clear advantage over other systems based on natural vesicles, which rely mostly on the EVs derived from genetically unstable cell lines. I would call our system as the first generation EV-based genome editing platform. It can be further improved to increase both safety and the efficiency. The vesicles can be engineered to contain specific proteins on their surface, which are then recognized only by a particular cell type or a tissue. It's like adding a specific key which can unlock only a certain door. Moreover, the packaging of the genome editing tools can be improved in order to increase their concentration in the EVs. This will lead to higher genome editing rates, which ultimately may cure a patient from a genetic disease.